All right. Thank you everybody and welcome to Reza's Thursday seminars on the cerebellum or Tuesday seminars on the cerebellum. My name's Alden and I'm a PhD student here in his lab and I'll be hosting today in his stead. I want to introduce our speaker today, Spencer Brown. Spencer is a postdoctoral researcher in Indira Raman's lab with a background in vivo neurophysiology and imaging. He completed his PhD in 2019 in the Raman lab, where he studied how cerebellar Purkinje cells and nuclear cells encode sensory motor input in the rate and timing of their action potentials. In 2021, Spencer joined Mike Hauser's lab as a postdoc researcher at University College London, where he engineered a technique to perform simultaneous 2-photon calcium imaging and holographic stimulation of Purkinje cells while imaging hippocampal CA1 neurons. In 2023, Spencer rejoined the Raman lab to continue studies on cerebellar spike synchrony using novel voltage imaging techniques. Today, he will be teaching us about synchrony in molecular layer neurons. Go ahead, Spencer.
All right, thank you so much for the kind introduction, Alden. I'm really excited to be here today to tell you all about the work I've been doing with Indira over the years, and especially about the work I've been doing with her more recently, where we've been looking at the activity of molecular layer interneurons during sensory-evoked whisking using high-speed voltage imaging. All right, so let's get right into it. Okay, so the question that really drives me as a neuroscientist is that I want to understand how patterns of spiking within populations of cells in the cerebellum and in the brain in general result in a signal that can impact behavior. Now, that's a very broad question, and we can break that down into two categories of more tractable questions specifically. So one thing I want to know is, how are sensory motor inputs transformed into patterns of activity at the level of individual neurons? And then subsequently, how are neurons of the cerebellum then communicating that transformed information to one another? And specifically, I have a great interest in understanding how information is being transmitted between the cerebellar cortex and neurons of the cerebellar nuclei. And so in order to understand how neurons of the cerebellum are communicating with each other, especially the principal cells, we need to consider one fact, and that's that Purkinje cells are highly convergent onto cells of the cerebellar nuclei. The convergence ratio that's been described has been on the order of 50 neurons to every single nuclear cell. And so this provides the circuit with a capability of transmitting information in multiple ways. One way that has been described is through a rate code. And I'm showing you that here in the middle. So since this is an inhibitory synapse, we know that if there's a population of Purkinje cells that suddenly increase their firing rates, this should lead to a reduction in the firing rate of their downstream targets in the cerebellar nuclei. Likewise, if Purkinje cells suddenly decrease the number of spikes they're firing in a certain window, this should lead to an excitation of the cerebellar nuclei for a certain period of time. So there's a lot of evidence that a rate code is happening in the cerebellum, and this comes from a number of studies where it's been shown that there are often strong correlations between population firing rates of neurons in the cerebellum and kinematics of movement. And this has been described for a number of behaviors, and I'll just list a few of them. This is not comprehensive by any means, but it's been described for smooth pursuit, the vestibulocular reflex, saccadic eye movements, conditioned eyelid closures, reaching, and whisking, to name a few. And I've listed some relevant citations below, but yet again, this is not comprehensive. I could fill this entire slide with citations. But that's not the whole side of the story. So another way in which neurons can encode information when there's a convergent system is actually in parallel on a millisecond time scale. And we call this the synchrony code. And so synchrony can occur really in two ways, either through the synchronization of the timing of the spikes of Purkinje cells, which can have an inhibitory effect on the cerebellar nuclei, or through the synchronization of the interspike intervals of Purkinje cells, which would have a disinhibitory effect on the cerebellar nuclei shown here. And these things are not mutually exclusive. You can have synchronous spikes followed by synchronous interspike intervals. And so what evidence do we have for synchrony? And so synchrony has been observed a number of times in Purkinje cells between their simple spikes, but also in MLIs because of electrical coupling. We also know from modeling studies and dynamic clamp studies in vitro that cerebellar output neurons are actually quite sensitive to Purkinje cell synchrony, and this is owing to the fast iPSC kinetics that are observed in cerebellar nuclear cells from Purkinje cell input. And lastly, and more recently, it's been demonstrated that synchrony in Purkinje cells in particular tends to be correlated with aspects of behavior and sensory input. And so it's been shown specifically that during the deceleration of movements, reaching movements and saccadic eye movements, that when Purkinje cells are reducing their firing rates, it's often observed, or it's been observed twice, that the leftover spikes are often synchronized on a millisecond time scale. And me and others in the Raman lab additionally have shown that when one provides a sensory input to the mouse whiskers, for example, we often find that there's not this synchronization of simple spike timing, but we find a synchronization of the gaps in firing of populations of Purkinje cells. And I'm going to tell you a little bit about that work now before we get into looking at molecular layer interneurons, because I think it provides some important context. All right, so when I joined Indira's lab as a PhD graduate student, I actually found it very hard to believe that synchrony was possible between biological neurons because it happens on such a rapid timescale. It seemed too electronic to me. And so the approach that I initially took in studying the cerebellum was to simply make single unit recordings from Purkinje cells and cerebellar nuclear cells during a relatively simple reflexive behavior. And then it turns out I eventually ran into synchrony along the way, but we'll get into that. So the behavior that I brought into the Raman lab was this sensory-evoked whisking behavior. And so what that means is when one gives an air puff to either the whiskers or the whisker pad of a mouse, what one will see is that the mouse will reflexively move its whiskers forward extremely reliably. Here's an example of what that looks like on a single trial over here, where the whiskers will be resting prior to the stimulus, which is a very brief air puff stimulus. And then following that, there will be a forward movement of the whiskers that lasts about 60 milliseconds long. And this initial movement and the response is very consistent across trials. And later on, you might get some rhythmic whisking that occurs, which is probably more voluntary in nature. The reason why I chose to study reflexive whisking in the cerebellum is because there's actually a large body of literature that shows that the cerebellum is intimately linked with the sensory system of the whiskers and the whisking motor system. So what is known is that sensory input comes in through the whiskers, goes through the trigeminal system, and the trigeminal system sends mossy fibers into the cerebellar cortex, primarily in lobules, Crus 1, Crus 2, but also in the paramedian. After being processed by the cerebellar cortex, this information is being transferred to the cerebellar nuclei, which has been shown to be directly connected to facial motor neurons in the facial motor nucleus, as well as directly connected to the whisking CPG in the intermediate reticular formation. And so we think that the cerebellum is a great place for looking at whisker-related sensory motor integration. All right. So when we give these air puff stimuli to the mice, what we see on average is, again, this reflexive protraction of the whisker movement, so if you take into account the latency that it takes for the puff to hit the face, which is about 6 milliseconds, you see this movement start at about 14 milliseconds after the tactile input reaches the mouse. When we record in cerebellar Purkinje cells, so these were single unit recordings made over many trials, looking at the responses of these cells to sensory inputs. What we find in those cells is that they have a basal firing rate of about 75 spikes per second. I've plotted that as spike probability on the y-axis. And then after the air puff at time 0, what we observe is initially this rapid suppression of Purkinje cell simple spiking. And these are 2-millisecond bins in this histogram. And so what you're seeing here is actually a 4-millisecond suppression of simple spiking. That's extremely rapid and brief. Following that, there's a secondary oscillation in the Purkinje cell response, and then a maintained increased firing rate that lasts for about 100 milliseconds or so. And we think that this is correlated with the position of the whiskers. Now, we can learn a lot from these single unit recordings of Purkinje cells when we observe this 4-millisecond simple spike suppression, and I'll tell you why. So essentially, so I'm going to go through a very quick exercise, and the exercise is if I have a Purkinje cell that's firing at 80 spikes per second, it's going to have a mean interspike interval of approximately 12.5 milliseconds. This is a simulated spike train, and the question is can you spot a 4-millisecond simple spike suppression within this spike train? And the answer is no, and that's because the response is actually briefer than an interspike interval. It's only when there's a population of Purkinje cells not firing all at once for this brief period of time that this response actually can be observed and actually can have an effect on the cerebellar nuclei. And so we think that this very brief suppression of simple spiking is evidence for Purkinje cells synchronizing to this sensory input, and that it's essential for transmission of these brief gaps in firing. All right, so in support of that, that this is a population code on a rapid time scale. When we look in the cerebellar nuclei, what we find is two things. So first of all, of course, they're firing at a relatively high level, so at about 70 spikes per second. And when we give that air puff, we see first two transient increases in spike probability, and then there's this late decrease in firing rate that lasts for some time. When we overlay that nuclear cell response on top of the Purkinje cell PSTH, what we found is that the nuclear cells are actually firing within the gaps in Purkinje cell firing. So especially during this early 4-millisecond simple spike suppression, but also during this later oscillation that we saw in the PSTH of the Purkinje cells, which makes us think that this is also being transmitted to the cerebellar nuclei. And so next, given that we saw this nuclear cell response, and given that it was relatively brief, we wondered whether or not it actually contributed to the whisking reflex that we are studying. And so in order to look at that, what we did was we suppressed activity in the cerebellar nuclei by stimulating Purkinje cells that were expressing channelrhodopsin. And when we do that, in fact, we do find that relative to control whisker movement shown in black, the whisker movements that occur shown in blue when we shut down the cerebellar nuclei are dramatically reduced in amplitude. And so we think this provides evidence that this synchronous suppression of Purkinje cells that is disinhibiting the nuclei is actually contributing to the generation of the whisker movement during this reflexive behavior. And now, so an air puff stimulus is actually not a whisker-specific stimulus, and it's not supernatural. And so what we wanted to next do, and this is from more recently published work, came out this year actually. What we wanted to next do is look at how these Purkinje cells in CRUS1 and CRUS2 respond to whisker-specific stimuli. And the approach we took to do that was to contact the whiskers with a moving bar. And when we did that, in fact, again, we see this rapid suppression of simple spiking that only lasts for 4 milliseconds. And it turns out, even when mice are whisking against the bar themselves, we still see this rapid, brief, simple spike suppression. When we look at the cerebellar nuclei, what we find is, again, there's this inverse coding that we observe, but it's on a very fast time scale, right? The largest nuclear cell response in both cases occurs during that very brief 4-millisecond window where Purkinje cells are not firing. One thing we also observed, though, which was interesting, is that during active contacts, when there's also a motor program going on, there are these early elevations in both the firing rate of Purkinje cells as well as the firing rate of cerebellar nuclear cells. And so we wanted to look a little bit closer at that. And so if we look at the active contact response on a much broader time scale, we notice a few things. So first, we notice that the primary place where there's this inverse correlation between spiking in Purkinje cells and nuclear cells is following the sensory input at time 0. They're anti-correlated here. If we look outside of that window where sensory input is coming in, and instead focus on the parts of the trace where there's changes in whisker position related to movement, we find that the relationship between spiking in the Purkinje cells and cerebellar nuclear cells is actually less consistent. Prior to the contact, nuclear cells and Purkinje cells are co-modulated, whereas after the contact, they're not. And so we think this is really cool. We've found a way to trigger this synchronous simple spike suppression using sensory input, and it seems to be extremely effective at engaging the cerebellar nuclei via this anti-correlated spiking. Okay, and so that really sums up the previously published literature. Now I'm going to talk to you about the work I've been doing on molecular layer interneurons in CRUS-1 and CRUS-2. And so we were motivated to look at what molecular layer interneurons were doing, primarily because we saw this rapid simple spike suppression in Purkinje cells. We wanted to know, are MLIs responsible for causing it? And it seemed likely that they are, given that they're the primary source of inhibition to Purkinje cells. And it's known also that when one gives air puffs to the whisker pad and records from Purkinje cells, or sorry, molecular layer interneurons in CRUS-1 and CRUS-2, they will find more often than not that MLIs fire spikes. And so, given this and our previous work on that suggested that Purkinje cells were synchronizing their activity, we wanted to ask the question of whether MLIs are also synchronizing their activity and whether or not this synchronous spiking that could be happening in MLIs is responsible for this 4 millisecond simple spike suppression. And further, if that is true, we wondered whether MLI synchrony might contribute to motor output. And so the approach that we chose to take in studying MLI synchrony is in vivo voltage imaging. And what's great about in vivo voltage imaging is we can use it to measure the spiking of dozens of MLIs on a millisecond timescale. And so now I'm going to tell you a little bit about in vivo voltage imaging. Well, voltage imaging in general first. So what is voltage imaging? So voltage imaging is an optical method for monitoring the membrane potentials of cells using voltage indicators. Those indicators can come in the form of dyes or genetically encoded voltage indicators. So I'm showing a diagram of a genetically encoded voltage indicator on the right, and how these molecules work is they have a voltage sensing domain that responds to changes in voltage across the membrane. When that happens, they change their conformation and then alter the fluorescence of a fluorescent protein that's attached to them. And as I said before, so what's really great about voltage imaging, it allows you to have both high spatial and temporal resolution when you're recording. So you can look at neurons on the micron scale across millimeters of tissue at kilohertz frame rates. And there has actually been success with using voltage imaging in the cerebellum. This has been shown in vitro by prominent labs in the field. And I was inspired by their work to actually go ahead and try to do voltage imaging in vivo of these MLIs. But historically, voltage imaging has been very difficult to achieve in vivo. And this is for a number of reasons. One being that the voltage indicators that are currently available produce relatively small signals, and that's not totally their fault. Part of the problem is that the molecules are restricted to the cellular membrane of these cells. Another issue with voltage imaging is that the signals we're trying to image are extremely brief, right? Action potentials are less than 1 to 2 milliseconds long. And so in order to capture them, you need an imaging system that is extremely fast. And then a third, probably less appreciated problem with trying to do voltage imaging, especially in vivo, is that the microscopes we've all invested in over the past 10 or 15 years are really tailored to calcium imaging. And so we need to use new imaging techniques to address this problem. All right, and so if one were to design the ideal microscope for voltage imaging, what would it be able to do? So first, you need to accept the fact that when you're voltage imaging, photons are scarce in space and time. And so your ideal microscope will have optics that maximize photon collection, photon detectors that are sensitive and have low noise, and be able to achieve, it should be able to achieve kilohertz frame rates. And a really good candidate for this is high-speed one-photon microscopy. And this technique has been pioneered by both optical engineers as well as the bioengineers who are developing indicators for one-photon microscopy voltage imaging. And so the approach that we're taking to do voltage imaging in vivo of MLIs is to use high-speed wide-field imaging combined with DMD targeted illumination. And so let's break that down into what that means. So here's a diagram of the microscope I'm using. It has a high-speed camera on it that's able to image from a field of view that is about 700 by 700 microns squared at an imaging rate of 2 kilohertz. So we can track action potentials. The other side of the story is that we're using a DMD, which stands for digital micromirror device. And if you don't know what that is, you've probably used one before in a projector. So these chips are used to project images. And so what the DMD is great for is it allows us, if you look over here on the right, it allows us to target light to individual MLIs in the cerebellar cortex. And it turns out that this technique is extremely important because it dramatically increases the SNR when you're using one photon microscopy, as well as it prevents tissue damage from the high light levels that are needed for voltage imaging. And you know, what's particularly great about this microscope is it allows me to record from 10 to 30 MLIs simultaneously with action potential resolution. Okay, and here's a zoom in of a couple of the neurons that have illuminated here. So what you can appreciate is, first of all, the spots of light that I've used, to target those neurons. And then you can actually see the neuronal membrane of these MLIs. And that's because they're expressing this fluorescent ASAP-based genetically encoded voltage indicator. All right, and so what do recordings look like from these MLIs when we use voltage imaging? And so here's one example recording from an MLI. That was performed at 3 kilohertz in vivo using an ASAP-based voltage indicator in the NOS1 Cree line. And what you can appreciate from this video is that, first of all, you can see the cell up here. This circle is the cellular membrane. You can see the cell blinking. Those are the action potentials. And what I've plotted below is the fluorescent signal from an ROI drawn around that MLI. And you can see that we can actually track the action potential firing in this neuron at high speed. And you can also see that we're able to track them despite their high firing rates, right? MLIs fire at 20 to 30 spikes per second. And so it's actually not possible to really capture what they're doing using calcium imaging. Okay, so speaking of calcium imaging, how do these signals that I'm imaging in MLIs stack up against the signals that one would see using calcium indicators? So what I'm showing you here is the spikes from the previous slide. And I want to point out a few things. First of all, the kinetics of the response here, you can notice that even though we're imaging at 3,000 frames per second, we're only really getting 3 or 4 data points per spike, right? These GEVIs are extremely fast. Another thing to appreciate, which is you'll be better able to appreciate it in contrast to the calcium signal, is that the amplitude of these spikes is actually relatively small. And so on a per pixel basis on the camera, we're getting about 6 photons at the peak of this action potential. Now, relative to, so this is really interesting when you compare these signals to what one finds when they do calcium imaging of Purkinje cell dendrites using GCAP8F. And so that's what one of those transients looks like, right? So the tau of the fastest genetically encoded calcium indicator, GCAP8F, has a tau of about 80 milliseconds. And so this makes GCAP actually not really great for tracking, the precise spike times of cells. But you can see that GCamp is great because the amplitude of these signals is so amazing, right? You can essentially use any microscope you want and capture these signals. And just to give you an idea of, again, the difference in the amount of photons we're collecting. So if I integrate under one of these spikes recorded with voltage imaging, I get about 15 photons per transient per pixel. Whereas if I use the exact same imaging parameters, get this transient and then integrate it under it for the GCamp, I get about 4,000 photons per transient per pixel. So there's orders of magnitude in difference in the amount of photons you get when you use GCamp versus voltage indicators. All right. And so the best thing about voltage imaging is now it puts us in a position to actually record from many MLIs or neurons in general at once. And it allows us to start to assess things like, trial by trial synchrony in these cells on a very fine-grained scale. So let's talk about that a little bit. Well, first, I want to say a few more things about what's great about this technique. So you can from voltage imaging, you get the spikes of the cells, but you can also get the XY locations of the cells, and you can see activity dependent parts of the membrane. So these on the left are all of the spike triggered averages of cells that I recorded from in a field of view. There's about 20 to 30 cells here. If we zoom in on a couple of those cells, which are plotted over here on the right, you can see the membrane of the cell. So this is the part of the cell that is active when there's an action potential. And then we also see these processes come off the cell body. And we're not really sure what those processes are. They could be dendrites or they could be axons, but we have reason to believe that they might be axon initial segments. What's even cooler, though, is when you look at these STAs, these spectral averages over time. And so if we do that, we can actually look at, where the spike is initiating in the cell and then watch it propagate outwards into the neural processes of the MLI. Okay. And so now let's get into the real biology here. So first of all, how do we know that we have MLIs? Well, we're using this NOS1 Cree line, first of all, which is supposed to label MLIs, but we also have, the morphology of the cells, the basal firing rates of the cells. And so when we measure the diameter of these STAs from the recordings, we find that the cells are approximately 11 microns wide, consistent with them being MLIs. And when we measure their basal firing rates, we find that they fire at about 30 spikes per second. Again, consistent with them being MLIs. And lastly, and really importantly, since this is one photon imaging, we can assess whether or not these are single units, as one has to do with recordings from multiple cells. And when we take the autocorrelograms of these cells, we find that there are very few spikes around time zero, suggesting that these cells that we're recording from are actually single units. Okay. And so as I had done before, I wanted to see how these cells responded to sensory inputs given to the whisker pad of mice. And so what I'm showing you here is 3 simultaneously recorded MLIs recorded across about 50 trials, although there are 100 trials in each one of these recordings. I'm just not showing them all. And what you can appreciate is that, the MLIs are responding. Another cool thing you can appreciate from these plots is that there appear to be correlations in periods of spiking. For example, during this period where there's a low firing rate in this MLI, it's also here and here. And then there are also some trials that you can observe where there's high levels of spiking, and this seems to be shared across MLIs. And so what's great about these recordings is we can look at them in multiple ways, right? So we can look at them from the perspective of just trying to figure out what on average individual MLIs are doing, or we can also look at what's happening on a trial by trial basis across multiple MLIs. And so first, I'm going to show you what's happening on average. So we've recorded 261 MLIs in this study. They all went through 100 trials of air puffs. And this is what their average response looks like. So following the air puff at time 0, we see this rapid increase in spike probability in the cells, followed by a second secondary increase in spike probability, and then this maintained increased firing rate in these population of cells. This was consistent on the level of individual MLIs in the population. So each line on this heat map is a 2 millisecond histogram from a single cell, and you can see that most of them are firing this double peaked response, followed by this maintenance of a high firing rate. What becomes really interesting is when we compare this population PSTH of the MLIs to the population PSTH of the Purkinje cells that I've shown you previously. And when we do that, what we find is the MLIs are firing at the precise time that they should be in order to result in this rapid suppression of simple spiking, which is quite cool. And so this made us think that they're likely responsible for generating this response. Okay. So that was about average responses of MLIs. We can also look at what MLIs are doing on a trial-by-trial basis on single trials. And so what I'm showing you on the left over here is a recording from 21 MLIs simultaneously. And what we're looking at is how they are all responding to the air puff and the relationship between the spikes among them. And so what you can appreciate from this trial is that after I give the air puff, there's this lining up of spikes across the MLIs. Now we can look at that a little bit more clearly when we plot the spike times as a raster, which is shown over here. And what you see when we do that is after time 0, when the air puff comes in, there's this synchronization of spikes in this population of cells. And we can quantify that synchronization using a very simple coactivity metric, which simply counts the number of spiking units in a 4 millisecond window. So when we do that, we find that for this particular trial, the MLIs, they have some basal level of coactivity, right? Because they're all firing spontaneously. And then after the air puff comes in, 16 out of 21 of those MLIs are firing a spike within a 4 millisecond window. And that is synchrony, right? And so we can convert that coactivity into synchrony simply by dividing the number of active units by the total number of units. And when we do that, we find that, in this trial, there is 76% synchrony. We can also look at the distribution of synchrony shown over here on the right. And what I have plotted is just the STAs of all the cells that are synchronizing on this trial, shown in red, versus the STAs of those that are not synchronizing on this trial. And, we've actually found that the amount of, the cells that are synchronizing does shift from trial to trial. Now that was,
Spencer, a quick question. Have you guys accounted for like firing rate in these kinds of measurements? Because
right. Do you mind if we defer that question until the end? Because I think that's a good discussion to have.
Great.
Yeah. Just remind me. Okay. So we can also look at what these MLIs are doing across, the hundreds of trials that we've recorded. And when we do that, and we look in the early window after the air puff, what we find is that, the level of synchrony, so this is now a synchrony metric. This is no longer coactivity, so we've divided by the total number of cells. But what you see is that there's variability in these initial peaks in synchrony. And that's better observed when one sorts the trials according to the peak amount of synchrony that occurs during that early time period. And when you do that, you find that there are trials that have approximately 60% synchrony, and then there are trials where there's not so much synchrony. And if we do that across all recorded groups, I looked at 17 groups of cells, and the group size is around 10 to 30 MLIs. We've got about 1,700 trials here. If we look at the peak synchrony across all those trials, we can get a peak synchrony probability distribution within this time window, 14 to 40 milliseconds. And what we find is that approximately on average, there's 50% synchrony on a given trial, although it can be lower and higher than that. Right? Some trials, there's 60 to 70% synchrony. And so what's really cool here is, we're actually able to measure gradations in the amount of synchrony, right? It's no longer a binary thing. And that's because we're recording from many neurons at once. And this motivated us now to look at what the behavior of the mouse is doing when we have this graded level of synchrony across trials. And so what we did was we binned that synchrony from that probability distribution into 10th percentile bins, and then took the average synchrony traces from those bins in the MLIs. This is self-imposed, but it looks like this over here, right? We have a gradation of synchrony. And now what becomes interesting is what we see when we look at the behavior sorted in the exact same way. And when we do that, we find that the evoked whisker movements tend to be related to the amount of peak synchrony during that early window, right? On trials with high synchrony, there are larger amplitude movements, and on trials with lower levels of synchrony, there are smaller amplitude movements. Now, if we plot the peak amplitude of this whisker movement versus the peak synchrony that we see in the MLIs, we find on average that there's a very nice relationship between peak synchrony and peak protraction amplitude. And so we really do think that these MLIs are synchronizing, and that it has probably, you know, a suppressive effect on the Purkinje cells, and then a disinhibitory effect on the cerebellar nuclei, and it's affecting movement in that way. And so that's the end of the talk. I'll summarize what I've taught you today. So what we think is going on in this relatively simple circuit is that when you give a sensory input to the whiskers, this gets transmitted to the cerebellar cortex. It engages MLIs in a synchronous manner. And then this results in a what we think is a synchronous simple spike suppression in Purkinje cells. This disinhibits the cerebellar nuclei, which can impact ongoing reflexive whisker movements. And I think importantly, when we look at diagrams like this in the middle, it's important not only to consider single cells, but rather to consider that there are populations of cells, and that this is a convergent network. And so information can be processed in parallel and at a rapid time scale. And if anything, I hope that you take away from this talk that synchrony appears to be a relevant variable to consider when investigating the cerebellar neural code. All right, and with that, I would like to acknowledge the people who have been involved in this work along the way, either directly or indirectly. So I would like to first thank, you know, the Raman Lab, especially Indira Raman, for taking a risk on me on this project. You know, she doesn't usually do imaging. I wanted to do it, and she invested, and that was great. I would also like to thank the members of the Raman Lab who have been with me along the way. I would like to give a shout out to Francois Saint-Pierre's lab, who provided me with the voltage indicator that I'm showing you in this study. If you have any questions about it, you know, give him a call. And I would like to thank the postdocs and graduate students who were involved in developing this indicator. And lastly, I'd like to give a shout out to the Heuser lab, where I learned everything there is to learn about cerebellar imaging. And I'd like to thank the postdocs, especially Dimitar, who introduced me to calcium imaging in the cerebellum, and Jacques, who really inspired me to do in vivo voltage imaging. And with that, I'll take any questions.
May I ask a question?
Yeah, absolutely.
Yeah. Thank you so much for the great talk. I was wondering if you checked for MLI type 1s and type 2s based on Bedreger's work.
Yeah, and so I have tried to look for MLI 1s and type 2s. I can't remember which one of those inhibits the other. I think it's the MLI 2s, right?
Yeah.
And
MLI 2 suppress MLI 1, and MLI 1, I guess, suppress the Purkinje cells, which is relevant too.
yeah, absolutely. Yeah. And so I have looked for like inhibitory cross correlograms between these cells. And I'll say I've found it a few times, but it's not very common in the region that I'm recording from.
Okay.
Yeah. But you know, there's a distribution of firing rates that we see in these cells. And I think the MLI2s are supposed to have lower firing rates. So maybe that's them.
Yes, I was wondering if you see like lower firing rates MLIs in the distribution. Yeah.
Yeah. And so,
and less synchrony probably between MLI2s than because they don't have the fat coupling.
Right. So yeah, we have not investigated this thoroughly, but we do in fact see some MLIs that are firing around 5 to 10 spikes per second, which is on the lower end. Yeah.
Thank you so much. Appreciate it.
Yeah, absolutely.
Beverly?
Hey, Spencer.
Hey,
How deep in the molecular layer do you think you're imaging?
so this is, about 50 to 70 microns deep.
Okay, I mean, that might be relevant to the subpopulations that you can access of the molecular and layer interneuron types.
Yeah, you're right about that.
Yeah,
Yeah,
maybe
and
more coupling in the deeper layers.
there might be, yeah. And
Yeah.
we do have interests in comparing, what the deeper neurons are doing relative to the more superficial neurons, because, in general, basket cells tend to be located deeper within the molecular layer. Yeah.
Thanks.
Yeah, absolutely. Right.
Dr. Raymond? We can't hear you.
Hi, Spencer. Can I ask you about timing and the expected time for inhibition or disinhibition to affect like downstream and all the way to the behavior? Because in your last set of results, you showed a pretty long delay between the peak synchrony and the MLIs and the effects on the whisker movement, which were like 50 milliseconds later.
Right.
And then there were some places where there were like simultaneous was the increase in firing in the nucleus and the decrease in the Purkinje cell, like so that there was like no delay at all. And so there are some places where I saw what looked like the expected latency and some places where there wasn't. And I wonder if you've thought about latencies and as a way to assess what could be causal and what might be not causal.
Right. Yeah, absolutely. We're thinking a lot about this. And so sorry, I can hear myself now for some reason. The other side of the story is that, so you're right in saying that a lot of these cells go off near simultaneously. There might, in some cases, there's about a 2 millisecond lag, which is what you would expect from a synapse, a synaptic delay. But in a lot of the cases, it's near simultaneous. And I think part of the explanation for why that might be is, like, for example, in the cerebellar nuclei, we know that there is convergent input from Purkinje cells as well as mossy fibers. And so this disinhibitory effect we see in the cerebellar nuclei is not only disinhibitory, it's also driven by excitatory input. And so it might be that the Purkinje cells are actually modulating cerebellar nuclear cell responses to mossy fibers. It's a combinatorial thing. And so, yeah, you probably need more than just Purkinje cell inhibition to get this, but we haven't directly tested that.
And what about the very long, like 50 millisecond delay between the MLI synchrony and the effect on the whisker?
Right. Yeah. And so, yeah, it is, that delay is very interesting. And we even saw it in our earlier studies when we looked at the cerebellar nuclei. It's kind of hard to see here, but right. So cerebellar nuclear cells are really only firing during the first 30 milliseconds following the air puff. And it seems like when we inhibit the cerebellar nuclei, we're affecting the movement on a longer time scale than that, right? These movements are being affected at around 70 milliseconds later. And so it's hard to really know why that latency is there, but it might have to do with how the whisker circuitry is organized, right? Like there's an inhibitory CPG that drives the whisker system, which can actually add delays to the onsets of movements. And so, yeah, it's an interesting question, and we can't fully explain why it's happening. Yeah.
Thanks.
Yeah.
Can I ask a question?
Yeah, absolutely. Hey.
Could you bring back the summary slide, please, that you just had in response to Jennifer Raymond?
Yeah, absolutely. So.
And the PSDHs.
Yes.
Yeah. So I was interested in the MLI versus Purkinje PSDH here.
Yeah.
And the interesting fact that they presumably both receive similar, or from the same population of parallel fibers, similar inputs. Yet the MLIs seem to win. They get to spike first, and of course they then inhibit the Purkinje cells first before the Purkinje cells increase their firing rate in turn. So I'm wondering what your thoughts are on why this might be. Is it because the MLIs are smaller and so fewer parallel fiber inputs suffice to bring them to threshold, and there's a lot of safety margin because many of them are brought to threshold almost instantly with the onset of the stimulus? That's what you show in your beautiful population recordings. And then it's just this mass of MLIs that inhibits the Purkinje cells. And Purkinje cells, of course, being bigger and maybe having a slightly more diverse set of inputs, taking a bit longer to reach threshold. What do you think about why this is?
Yeah, so your question is essentially, yeah, why does inhibition show up first in the Purkinje cell, right? Despite, excitation probably reaching the Purkinje cells at the same time. And this has actually been an active area of investigation in the Raman lab for 4 or 5 years. And so I'll tell you about that now. I brought a slide with me. And so the reason why we think inhibition wins out first, with this sensory stimulus is because there is synaptic facilitation in the parallel fiber pathway. And so, for example, if you look at this whole cell recording from a Purkinje cell in slice, where we're stimulating, parallel fibers in 50 hertz trains, what you find is that if you look at the excitation in a Purkinje cell from the parallel fibers, at the first pulse, the excitation is relatively weak. After multiple pulses, it increases in amplitude. If you look, conversely at the inhibition, what you find is it's relatively stable throughout the entire train of stimuli. And so what we think is happening early on when Purkinje cells are receiving a burst of inputs, probably both MLI and parallel fiber input at the same time, we think it's that the excitation is initially weak. And so inhibition wins out. And then later on, facilitation kicks in, and then you can cancel out the inhibition in a way.
And is the short-term plasticity of parallel fiber MLI synapses similar? Are they similarly facilitating, or is the first one already strong?
So the first one, I think they're facilitating. I don't know this literature as well, but what I do know is that MLIs are extremely responsive to granule cell input. They'll respond to even a single quanta of input.
Yeah, the input resistance is much higher. So you need fewer inputs / you probably have with this given sensory stimulus that you've applied a much bigger safety margin towards making the MLI spike compared to making the Purkinje cell spike.
Absolutely.
In addition to what you just say here, it's very, very interesting. So it's wired up to make the interneurons win and inhibit the Purkinje cells first, and that triggers, a 2018 paper. What you described in your 2018 paper, the very short notch in Purkinje cell firing.
Exactly. Yeah, that's
cool.
That's precisely it.
Thanks a lot. Cool stuff.
Amazing question. Thanks. Okay.
We had a question in the chat from Maxime. He asked, did you image in other cortical lobules to monitor the anatomical specificity of the responses that you see for those imaged MLIs, but also for your former EFIS studies? Put differently, what fraction of the cortex flashes in response to whisker pad puffs?
Right. That's a great question. And so, yeah, I've spent most of my time looking at CRUISE 1 and CRUISE 2. But I will say that in a few of the recordings I made during these studies, we did look at what was happening in lobule 6 of the vermis. None of those data are reported here because they were kind of one-off experiments. But what I can say is, yeah, in fact, there are neurons firing in the vermis as well, molecular layer interneurons, when we give the stimulus. I did notice, and I'm not really sure if it's totally true, that there seems to be less synchrony occurring in that lobule, right? It seems like there's more, it's really what we see is more of a slower, broad increase in firing rate in the MLIs. But, that needs to be corroborated with many more recordings. That was a one-off experiment. But yeah, of course, the air puff stimulus is not a whisker-specific stimulus. It engages kind of a full body reflex. One of those reflexes is the whisking reflex.
Hey,
right.
Sorry, I guess there was another question. I'll wait, but I want to ask you about synchrony.
Yeah. No, I think we can talk about synchrony.
There was another raised hand, I think. Looks like Jennifer, who was before me. I'm happy to wait.
You first.
Okay, thanks. Yeah, so I was just wondering how you think about the synchrony versus kind of like synchrony in yeah, how you think about synchrony in the context of changes in firing rate, and also the fact that really the sensory stimulus that you're presenting is synchronizing in a way. And so
Right.
what you have is like equally, you could have precisely timed spikes in individual neurons, which are in effect kind of creating a situation where the cells are spiking at the same time. Because it also looks like in slide 29, the one that you have your cursor on,
This
it also
one.
looks like you have changes like in synchrony prior to time 0. So that I think is just changes in baseline firing rate, right?
Yeah.
Like
Yeah.
it looks like your trials with more synchrony, as you're calling it, are
Right.
situations where your cells are more excitable. And so do you think that's an excitability thing or a synchrony thing, actually, like synchrony in the sense of, I don't know, some population state that is like distinct and meaningful?
That's that. Yeah, there's a lot to unpack there. Where should I start? Do you want to, sorry, yeah.
So baseline rate.
Baseline rate.
Yeah.
How do we think about it? And why is it related to what's happening on later in the trial?
Yeah, do you think it's, yeah, so maybe the first place to start is rate versus synchrony. So
Rate.
those things in the way that you're measuring them are, I think, conflated.
I agree. I agree that to some extent they're the same thing, right? Because ultimately what synchrony is on a short time scale, it's just an increase in spike probability, right?
No, it doesn't have to be though. It can be an increase in spike probability. It can be well-timed spiking without a change in rate. And that's like actual synchronous spiking. Like,
So how would that look to you? It would be a rapid increase and then it would go away.
well, it would look like something like this without a change in actual firing rate.
Well, I think it's hard to find that perfect type of synchrony because multiple things are going on here, right? Like we have the sensory input, which is, like you said, is likely engaging many mossy fibers at once. And this is going to, just by the nature of it, synchronize cells because they're getting the same input. And I think that's interesting. And that is what we're describing here. But, the later parts of this response are likely being driven by motor commands. Because we know these cells receive whisking-related motor commands, and that's going to result in an increase in firing rate. And maybe the reason why these two things are correlated, right, in the basal part of the response, as well as during the later part of the response, is that, like you said, there's some increase in excitability in these cells on those trials. It might have to do with, whether or not the mouse was whisking a little bit beforehand, whether or not the mouse was attending to the input that's about to come in. I don't really have an answer for that. It's a very high level question, but it's something we're interested in investigating. Yeah, I mean, did that address some of your questions?
I'm coming as a fan, so we should have a chat.
Yeah, no, I think it'd be great to chat about this. Yeah. Absolutely.
Okay. Thank you.
I guess I would just plus one Dimitar's point that it would be very useful, I think, for the field if you carefully distinguish what is like a very rapid rate code. So when Hannah Payne was in my lab, she was looking for evidence that like the interspike intervals in Purkinje cells might carry more information than just the rate, the population rate. And she found no evidence for that, but did find evidence that the Purkinje cells population rate is getting read out downstream on a time scale of like 3 or 4 milliseconds. And so, I would be very interested in knowing if you see evidence for something beyond the rate code in the Purkinje cells, or the MLIs seem like a place where it really might be there. And either answer is interesting, but if you don't account for the rate code, then it's hard to interpret. Like it would be really nice to know what is a rapid rate code of, the response to the input versus something that like is being intrinsically generated. I don't think you need the perfect condition. It is possible to have a synchrony event without a change in rate. But the standard analysis would be to take into account the population rate and look for synchrony beyond that. And I think that would be a really nice contribution here.
Yeah, absolutely. We can try to loop that in, a detailed analysis of rate and how it relates to coincident firing in neurons. So one thing I don't fully understand though is, so what is the difference between a rapid rate code on the timescale of 3 to 5 milliseconds and synchronous spiking within the same population of cells, with jitter of 3 to 4 milliseconds or 5 milliseconds? Like, is that just a difference in the way we're talking about the same thing, like increased spike probability across a population of cells, or is there something actually intrinsically different about those two things?
Yeah, I think the question is whether the circuit is generating synchrony beyond what you would expect from an increase in firing rate, because when cells increase their firing rate, their interspike interval will decrease from 12 to something shorter. And if it's stimulus driven, that could be well aligned between cells. And so, that's trickier looking at the onset versus in that sustained period, you could look for synchrony beyond what you would expect from the increase in firing rate. Yeah, so and it's sort of synchrony carries with it this mechanistic implication, right? Like it are the MLIs trying to fire together beyond just kind of, taking their inputs and thumbing them and doing their own thing. Are they somehow coordinating? And we know they have gap junctions, and how big of an effect is that? So I think it really is different. And we, using the word synchrony to mean things that cannot be accounted for by the population rate would be helpful.
Okay, yeah, absolutely. And maybe we can talk about this further at SFN if you're around. Yeah, great.
Hey, I have a question as well.
Sure.
That was great. I really enjoyed it. Can I ask a question about the previous slide where you show synchrony across trials?
Sure.
Looking at the figure on the left, it looks like there's a lot of synchrony in the first 10 trials and almost no synchrony in the last 10 trials. Is that, first, is that true? Is that generally true, or just in this image here? And if so, do you think that that is some kind of learning that's happening in the cerebellar circuit itself? Or is this some kind of habituation to your air puff over time, that it's whisking less? And I guess I asked that because it makes me wonder what the direction of causality is on the next slide. Are the animals whisking less? Are they just habituating and so there's less synchrony? Or is it the synchrony that's causing less whisking?
That's actually, yeah, that secondary question about the behavior is actually really interesting. And I don't think I've looked at that yet. And so I'm probably going to do that after this talk. But what I can tell you is we have looked at how the most, synchronous parts of the response change across trials. And in fact, there is a slight decrease in the amount of synchrony across trials. Whether or not that's related to the behavior is super interesting. And yeah, like I said, I will take a look at that, but I'm not actually sure. And I don't necessarily know the mechanism, because as you say, so this input we're looking at here is, so the response we're looking at here is likely driven by input from the trigeminal system, and the trigeminal system itself might be, desensitizing to the input. Yeah, so these are all really interesting questions. I don't have the answer yet, but yeah, what I can tell you is, yeah, there is a slight change in synchrony across trials.
Thanks.
Great.
I can't figure out how to raise my hand, but I just had one comment in the context of Jennifer's and Dimitar's questions about, is it really synchrony, and wanting synchrony to be something that sort of magically happens independent of outside stimuli or not. And my comment is a little historical with respect to my own lab. The reason we in this lab became interested in the idea of synchrony in the first place was the question of whether the cerebellar nuclei could distinguish when Purkinje cells were firing synchronously or not, regardless of what that source was. And the biophysical answer was yes, that if you have a synchronous firing of afferent Purkinje cells or a synchronous disinhibition by Purkinje cells, you could get one heck of a cerebellar nuclear cell output. And we're interested in knowing, is that ever engaged computationally by the brain to generate cerebellar output? The answer that Spencer has is yes, that there are circumstances when Purkinje cells will simultaneously shut off, probably because of molecular layer input, interneuron input, and that can lead to a very large response in the cerebellar nuclei. And so from that point of view, that's the level at which we were defining synchrony. And these other more abstract and theoretical definitions of, how that relates to rate. Rate still has a denominator of time. And if you make that denominator small enough, then you're going to get synchronous firing, right? And that's what we're actually observing. So I think that there are other ways of thinking about synchrony in a functional sense of how there's a readout through the cerebellum that may be somewhat orthogonal to some of the other definitions that have historically been used in various contexts. So I just wanted to add that in there as a way of thinking about synchrony.
Thanks,
I
Indira.
understand your, sorry. I understand that, Indira, but like if you use the word synchrony to mean something different than kind of what the broader field has used it to mean, then it kind of muddies things. Whereas population rate code is like, everybody knows what that means. The population rate increases or decreases over short timescales. And because so many people have taken synchrony to mean something beyond what you can account for by the population rate, it muddies things to use it to mean something different.
Well, synchrony means things happening at the same time, and we're careful to define what we're talking about. And so we've tried to define that for a period of 13 years now, that we're talking about Purkinje cells firing simultaneously or ceasing to fire simultaneously, which is a fairly standard definition of the word synchrony, both in the cerebellum and beyond it. But in any case, I guess we're at time.
All righty. Thank you so much, Spencer, for the wonderful talk and for the great discussion. We'll see you guys next week. Have a good one.
All right. See ya. Thank you.